So I was an engineering major at Berkeley, kind of got into biology through the simulation of biological systems. Went to MIT for my PhD, where I continued along those lines of research until I realized that we really had no way to rationally interact with biology in any reasonable way. So our ability to measure anything, our ability to perturb and manipulate the systems was really lacking in comparison to our ability to build models of those systems. So the models ended up being somewhat useless. And so what I kind of started switching to midway through my graduate work was building out essentially a technology stack around biology that would allow us to more rationally interact with biology. So that started with work at MIT around building some of the first synthetic viruses to kind of make these viruses that we were studying more like the models we were generating. I went to help start a company in the biofuel space, so engineering blue-green algae to make biofuels at the time. Came back to Harvard, worked on more base technologies around some of the stuff I'll talk about today. And then in around 2013, moved my laboratory to UCLA, where we've been kind of building out a lot of what I'll tell you about today and stuff at Octant. So I didn't prepare a ton in the sense that I'm hoping that you guys have questions and discussions. And B, I'm going to start more open-ended than Octant and kind of how I see what have been a series of massive revolutions in biology and biotechnology, and how I think that is playing out right now, and at least how I think about it. And I think there's going to be, so whether or not you guys are interested in biology or not, I think it's going to affect you over the coming few decades. I think being conversant and knowledgeable about, I think that how that landscape is changing is super important, just as citizens. And then more broadly, I think there's a lot of useful and really, I think right now is the right time for thinking about more serious computation around biological systems and how we build those things to interact more rationally with medicine and other parts of biology, and in particular synthetic biology. So, does that sound good? Anything before we start? Okay. Okay, so I will basically walk through 3 sets of technologies. The reading, writing, and editing of DNA. It's kind of just one lens and how to think about it, but I think in each of these base sets of technologies and how we manipulate the genetic code and how we understand the genetic code have been massively changing in biology over the last decade. And the scale and pace of that change is just really hard to comprehend. You probably have heard some of this before, and you've seen it from just what's been going on in the world. But this graph has been shown a lot of times. I just pulled this down from the NIH website. You can go to it. It's the cost of a raw megabase, so 1 million base pairs of sequencing costs. So 2007 is when I finished my PhD. And you start seeing this precipitous drop. So for a long time, we were basically matching Moore's law with sequencing. So you guys all should probably know about Moore's law. But the interesting thing about Moore's law is that it set a pace for discovery that also set a set of business models around expecting that pace to exist. And it's what led to this kind of combination in the development of programmers always expecting the next set of hardware-based improvements. And so you were able to be anticipatory around building the next set of apps over about a 50-year period. In biology, what happened in 2007 is we went through 35 years of Moore's law progress in approximately 5 to 7 years. So imagine being a programmer in 1985 and then showing up today and then saying, here are today's computers, what are you going to do with it? And realistically, what most people would do with it is like, make Pong faster, right? You wouldn't go and develop Facebook immediately. And so I think what we, both from the biology standpoint and from a technological standpoint, what you see today is a lot of, we're going to sequence all the things, right? We're going to sequence everything and infer the complexity of biology from that sequencing data sets. And there's been a lot of interest and excitement. I think this actually has really profound implications in society. So I often talk about some of these things, but and we were just talking about this before, but yeah.
Where did the cost go up in 2016?
Yeah, I don't know. This is just raw data from NIH. This is almost completely dominated by a company called Illumina that sets basically the lowest price on sequencing. This spike might have been due to the first production of the NovaSeq, which increased scale but also cost. And then this is a subsequent flow cell. So this was around the development of the HiSeq 4000 and NovaSeq when those started having some issues. But like basically Illumina has no competition on cost right now. So you could reduce this pretty easily, I think. It's just, there's no really market need to at this point. Yeah.
My understanding is that the sequencing has some sort of error bars around it in terms of accuracy.
Yep.
And I'm curious if that accuracy has been sort of kept the same across all this, or if it's, let's say, dramatically up or down within that.
I think for the most part, it's pretty similar. There's another axis, which is length, which is probably more important in the analysis of full genomes. And so, like, to this day, we don't have a complete human genome sequence, right? Because really the problem and the computational problem is around, like, how do you turn these short reads into something that's a full genome, and half or more of your genome is just a bunch of repetitive elements. So you can imagine how that would be super hard to kind of stitch together the full genome. So there are issues like that, absolutely. The bigger problem is we don't know how to interpret most of those things anyways. There's different kind of levels of products that people think about, either full genomes, but exomes are kind of one of the big things. And we can go into that in more detail if you guys are interested. But like the example I was going to bring up is that, and we'll talk about CRISPR and editing and DNA synthesis, but we were just talking about this before. I think sequencing alone has really big impacts. And I don't know how many of you have children, but if you do, you're likely to have had a prenatal test during the early stages of pregnancy that looks for a set of about, right now the standard is around 8 classic genetic diseases, around a class of rare diseases that result in non-disjunction. So this is an extra copy of a particular set of chromosomes or big portions of chromosomes. So the classic example here is Down syndrome. So in the 70s and for most of human history, about 1% or so, although I think it increased for a while because of the age of parents got older, but about 1% of live births resulted in Down syndrome. And the previous state of the art was mostly amniocentesis, which wasn't great at telling whether or not there was one of these events, but it was a super invasive test. And now, because of the discovery around fetal DNA actually floats around in the mother, that you can actually just do a blood test at around 6 weeks and tell pretty accurately if the baby will have Down syndrome. And by pretty accurately, it's like it gets most of the cases, but 50% of the time are on order. It's correct. So okay, so you have a test. What does that mean from a societal standpoint? What that means is in places like Europe, in specifically places like Denmark and Iceland, the rates of elective termination in those states or in countries are 99% or higher. So last year was the first year there were no Down syndrome cases in the entire country of Iceland. In the US, it's closer to we don't track as well, so it's around 70 or so percent is the best known estimates, choose to electively terminate. In most of Europe, it's north of 95%, 90 to 95%. So what's happening to the Down syndrome community in particular has been a big community because it was the largest form of mental retardation in human diaspora. And they built a pretty strong advocacy group around the rights and disabilities of people with mental disabilities. That entire population is being decimated or worse, right? Like that cohort of people that always existed will no longer exist. And maybe you agree with that or maybe you don't, but like I think the more interesting thing is over the course, what's happening now, I'll skip over some of this, is stuff like this. So if you look at websites like Good Start Genetics, these are actually, got cut off. But this is just a bunch of rare diseases that you might or might not have heard about that are starting to come online. And there's now hundreds to thousands of these, depending on how much you're willing to pay for the pre-carrier screening. So there's a few different types. This is I think if you saw this website, like in a sci-fi thriller from, you'd think, oh, look at this dystopian Good Start Genetics, prepared for, anyway. So this was started by a friend of mine, by the way. So like, it's not to, but in general, there's like carrier screening, which is before you have kids, you sequence yourselves and you can just see if you have like rare mutations that lead to the loss of function of a gene. Those are usually the causal agents for things that are called rare diseases. So rare diseases are usually caused by a single large effect mutation. You might be a carrier in the recessive form, but if your partner is also a carrier in the recessive form, then most of you have taken some biology as the Punnett square. You end up with 25% of chance of having a baby with both copies missing, and that usually leads to the formation of what's called rare diseases, and there's thousands of them. Usually because you're missing one of the genes that's not essential for life, but has severe consequences usually. And in general, there's 20,000 genes in the human genome on order, and so there's a lot of syndromes. Pre-implantation, so if you get a carrier screen, the next step is usually pre-implantation, where you select embryos that you want to actually screen you select embryos that you actually want to take forward into the pregnancy. You implant embryos, but you can select which ones you actually want to implant. This is getting increasingly the Wild West, for instance. So there are companies that will test not only rare diseases, but hundreds to thousands of other things, including the latest rage, which are called polygenic tests, which can test for more things other than super severe diseases, and we'll get into that in a second. And then obviously prenatal screening that I already mentioned. Okay, so that's sequencing alone. I'll mention just a couple of other things, and then we'll get to, why isn't that working? Oh, yeah, this just keeps on going, sorry. So writing. I will say, the ability to synthesize DNA, we think of as a tool to basically, so the genetic code, for the most part, contains most of the information necessary for life, and is also responsible for the genetic basis for a ton of diseases. Obviously, if you smoke, there's environmental causes to diseases and traits, but things like genetics actually play a very large role in your susceptibility to disease, even given environmental conditions. I think, did you have a question? Sorry. No, sorry. I think the general approach is the ability to synthesize DNA gives you a way to test essentially the causal mechanisms by which these things are occurring. So if I change the code and something else happens, typically I'm able to make inferences on what that mutation might do in the future. And so we're pretty interested in building synthetic DNA towards the end of not only building synthetic life forms, but also as a way for causal discovery of mechanisms in biology. And in fact, that's what a lot of my lab focuses on. We will talk about this in a second, but the big sets of technologies that really hit the cost curve here was the development or what are called microarrays. So this is a project by a company up here, called Agilent Technologies. It's a spin-off of Hewlett-Packard. So they took literally the same CMYK inkjet printheads from, and instead of CMYK, you have ACTG, and then you can actually do chemistry on surfaces, and you get these microarrays on demand. And each one of these things are basically a built-up piece of DNA that's 200 to 300 nucleotides long, and you can specify that with arbitrary precision. You can build millions of them for, depending on what they'll sell it to you. You can make it for super cheap. They'll sell it to you for a few $100. What we do is harness this as a way to get basically DNA, but with programmatic control. That's what, you get the image. Okay, so we've used this for all sorts of things. A lot of what we do is kind of like, let's change this piece of DNA and then look at the consequences of them in each cell. And this is a slow process. It's done one at a time. What we work on are methods to increase the scale at which we can build DNA, measure the consequences in a single experiment by a single person. We also do kind of fun things. So we've done the opposite, which is take arbitrary digital information and code it in DNA. So we've done stuff like, this is a paper from 5 or 6 years ago where we took these kinds of processes. That's my hand. My nails are kind of gross. But where we can code essentially megabytes to hundreds of megabytes. That's basically the scale we're at right now in incredibly small strands of DNA that can be stored in 3 dimensions and are stable over tens of thousands of years. So we did this for a book. And you can see, like, information density-wise, we're ridiculously information dense, but obviously not at a scale that current storage devices are today. So there's a bunch of groups now working on pushing this as a way to store information for long-term digital storage. And that's that work was basically super, super simple. We just took an ASCII encoded book, or actually it was just a PDF, and just took the bits and converted them into a fairly simple code around, in this particular case, G's and T's are 1 and A's and C's are 0. We can create short strands, and along with addresses that identify where the address of that thing is and redundancy. And then you can just synthesize this on the same inkjet printer and then make, nearly infinite copies by a simple PCR reaction for free. So this is the kind of stuff that we can do today. Yeah.
So what prevents you from going in that direction? Like, does it break or what?
There are people doing it. I mean, it's just like, this is a compressed graph in log 10 encoded. So like you're talking 2, 4, 6, 8. We're 10 to the 7th approximately off from current cost standards, right? And there are people working on it, and I think there are ways thinking about that, but it's like going to take a lot of development.
So is that cost related as well, or?
It's cost related, I think, is the biggest thing. Like algorithmically, it's just like there's various channels for, I don't know how many of you guys work or think about storage, but there's like channel codes for thinking of any particular medium. Like even, so these problems of repeats that happen in sequencing is a problem both for synthesis and sequencing, but it's also a problem for like storage medium like CDs and DVDs, right? So like, for example, most CDs that you have a channel code called EFM, which is called 8 to 14 modulation. That takes every 8-bit piece of information and converts it to 14 bits, mostly to avoid the series of repeats that happen when it loses registration from a series of non-pits in a CD, because you're basically relying on speed. So there's a whole bunch of like channel code development in this space, but also kind of hardware, software development around building much, much cheaper versions of this, mostly using and thought processes around archival storage.
So is this just a question of time based on Moore's law that we're going to store information on DNA?
I'm not betting my career on it. But like I will say that for me, the reason why I stopped working on this work, so this was some of the first work in this space, the reason why I stopped working on it is 2 reasons. One, I think the challenges aren't biological, but really manufacturing and scale. And I think if you look at current chip fabs and storage fabs, there are billions of dollars in investment on engineering. If you put that much investment into the engineering of this, I think you could make something for archival storage. But like, A, it's a storage problem, and I want to spend my life working on biological problems. So it's kind of one of the things that I've just decided that I'm not going to do with my life. But I think there are so Microsoft has a big collaboration with the University of Washington working on this. IBM and Texas Instruments have some stuff, but it's super early stage. It's like race track memory or like positioning xenon atoms on a surface. Like they're research projects right now. Yeah, it's incredibly slow. Yeah, but I don't know how many of you guys have seen like these. They're getting faster, right? Like if you look at nanopore sequencing. Do you guys know what nanopore? So nanopore sequencing. So these sequencers that you buy, we just bought one of the latest Illumina sequencers this last week. They're big. They're like this big. They sit on a desk. They're basically based on massively parallel imaging and flow-based chemistries. there's new, I think probably the future of sequencing is a new set of technologies called nanopore. And nanopore works by using a biological pore that sits in a membrane, and I probably have slides on this, but sits in a membrane. You have a biological pore that's about the width of a piece of DNA, and DNA's charge is negatively charged. So if you put a charge across that membrane, if you guys remember physics, it'll drive a force through it, and the current that's allowed through that gets blocked by the DNA going through, and it gets blocked in a sequence-specific manner. So you can monitor conductance through the pore as a way to relate the sequence going through to the sequence of the DNA that's being threaded through. So all of a sudden you have a single molecule device, and this actually, like the current Oxford nanopore, do I have internet? Oh, I do. Let me just try to get this to there. Oh, sorry. I tried to download Skype for Business and couldn't. I don't know. I didn't know it was different than Skype today. We had a Skype. Apparently, everyone in pharma in Europe uses Skype for Business. Maybe Oxford. So this is the current kind of deployed stuff. Images. It's about that size. You can kind of see it in the palm of your hand, and it plugs into a USB port. The newest ones that were just released, I think, are called the Flongle.
Wait, that sequences DNA?
Yeah, on the order of like these kind of old, newer machines. The Flongle, where's the Flongle? Anyways, it's much smaller. It's like this small. And it can sequence like, yeah, a megabase of DNA. No, I think that's the MinION. Anyways, they have the GridION, the MinION. Like, there's ones that are bigger that can sequence much bigger things. But the big thing is that they're super fast. They're real-time DNA sequencing, because you're just reading conductances in an array of nanopores. And there's no preparation, so you can actually just actually get real-time DNA directly on it. Yeah.
Is this DNA that's already sort of uncoiled and unmethylated and just like sort of totally pure? Or is this like real?
Yeah, so there's a whole
DNA?
bunch of things around. You're pointing to that this is chemistry, and DNA is not just DNA, but they're modified in all sorts of ways. And then there's RNA and
it's
other
all
polymers.
Yeah. And if it's in a chromosome that's all like chromosome together, then that
Usually the prep to remove proteins is pretty easily, and those kind of coilings are done by proteins. There's actually enzymes that are also in the mix that kind of separate strands. And there's a bunch of kind of startups in the space, but Oxford is probably the leader here.
But basically, the pre-processing happens, and then you put it in this machine.
Yes, but they're working on ways to basically get them
Avoid
directly
that.
from like blood extracts. And
So you can literally put in like a cell in it, or like put in a nucleus.
yeah,
or
so I have a
something.
friend that's starting a startup that's like using these to do real-time forensics from crime scenes and stuff like that. So there's like a whole bunch of stuff. The funny thing is on the synthesis side, it's like pretty trivial for me to like synthesize all the DNA needed to fake a DNA profile too. So like those are also happening in real time. We thought it would be our product to fund Octant in the early days, sell crime scene. Yeah, the future will be weird. So, okay. Did I just close my presentation? How do I do that?
Can you talk just a bit about why this doesn't, in the near term, replace the Illumina sequencer?
Yeah, I think it's scale. Basically, they're not at the same scale yet, but the pace of development is really fast. So there's definitely, I think, going to be a race, but like there's an entire phalanx of informaticians and computational biologists and software engineers that have been built and developed on short read sequencing based on Illumina. These long read sequencers that are based on Oxford are just much more error prone. And so it's going to take a whole set of equivalent people that start to develop those things. But in the end, the danger is that Illumina can always drop its price because it's actually cost basis for a lot of this stuff is pretty low. Okay. And then lastly, I'll just talk about one thing, and you guys have heard of this before, and I'm sure have seen talks around CRISPR-Cas9. It's their suite of new technologies around the ability to take synthesis and sequencing and just basically start instantiating that into organisms that we care about. It's always been like, for my entirety of my career, trying to engineer a particular cell, whether that be E. coli all the way to humans, has been some of the most challenging parts. And there's been a suite of tools, not just Cas9, but also TALEN, serine recombinases, base editors. There's just been this huge kind of explosion of a set of technologies that allow us to engineer cells in rational ways. Obviously, you probably have heard of gene editing. This is some of the first kind of clinical trials around an older set of technologies where we're just inserting a gene rather than editing the natural gene. This child was born with, you probably have, if some of you are almost my age, where you've seen The Boy in the Bubble. Do you guys remember that movie? It's a documentary. It's a disease called SCID, where you basically lose your entire immune system. And these, for a long time, the treatment were like putting people in literal bubbles that would completely isolate them from the environment. This is just to, usually the disease is caused by a deletion of a particular gene. It's a rare effect, large effect mutation, and you can reintroduce the gene. And this guy, Don Cohn, has spent his entire career trying to fix these babies, and the first 18 babies in clinical trials were all cured, all 18 of them. Yeah, yeah, for children, yeah, so alive babies, not embryos. So this has been a culmination of about 25 years of work, and my colleague at UCLA, Don Cohn, there's been a number of, he's also working on sickle cell and a bunch of other of these diseases that lead to loss of effect mutation. So you don't have to go and like CRISPR edit the change, you can actually just go and bring in the gene exogenously. The first, CRISPR? Or, yeah, I can. For a long time, what was the problem was that so the way we insert genes into mammalian cells, for the most part, for everything that's not in embryonic stem cells, so like actual human, is often through the use of viruses that we have re-engineered to basically serve as vectors of our DNA of interest. The first set of things were around a set of viruses called the adeno-associated viruses, which would lead to basically massive immune system responses and have issues around delivery. The newer generation of therapies are around a set of viruses called lentivirus, which is basically a gutted HIV genome. So these are basically HIV that we've removed all the bad parts and then can deliver a gene. There's a limited packaging length, so you can only deliver a certain size of DNA, and it integrates randomly into the genome. But for the most part, in cases where this type, so why does this work? Why this works is because it's an immune disease, and you can replace your immune system. And you can replace your immune system by basically irradiating yourself through chemotherapy or radiation therapy, and then reintroduce stem cells through bone marrow transplant. So the way that these kinds of gene therapies work is that you take bone marrow out, you find the stem cells, you engineer those stem cells, and then you irradiate your whole body to get rid of everything else, and then you put in the stem cells. And then that repopulates your entire hematopoietic lineage, which includes your stem cells. Includes your entire immune system. Does that make sense?
So over time you have like both the old cells and the new cells?
No, you pretty much only have the new cells because you've irradiated all the stem cells through radiation therapy. It's also why you see all the new gene therapies are always eye diseases or immune diseases for that reason. So Sangamo is a company also here that developed like the predecessor to CRISPR technologies called zinc finger technologies that was invented something like 20 years ago. And they've been working on bringing a gene therapy against HIV, which is basically the deletion of a gene called CCR5. And CCR5 is the entry point for HIV. And what they discovered, actually the discovery was super interesting and accidental. There was a patient with leukemia and HIV who got a bone marrow transplant for the leukemia, and it accidentally cured him of HIV too. And so this luckiest person in the world, basically they looked at the bone marrow transplant and it turned out it was a CCR5 null, meaning they had 2 missing copies of CCR5, and it turned out this was the co-receptor that HIV uses to get into immune cells. So if you're missing it, there's plenty of people walking around without this gene, and in fact, there's a lot more than you would otherwise expect because it's been a recently selected allele in the population out of Africa, mostly because heterozygous loss of function leads to less susceptibility to malaria. And so it's been a positively selected trait to lose the function of this gene in Africa. But it also so that's kind of a long story, but sorry, that's
technical limitation to
immune diseases.
Is there a technical, not ethical, but technical limitation to inserting arbitrary genes through the bone marrow?
Yeah, people were worried about people are worried about just randomly mutating a cell might lead to cancer, right? You can hit a what's called tumor suppressor gene, and that could lead to cancer. They haven't seen that yet with the newest generation of lentiviral vectors. So for what it's worth, that was a lot of the concern early on, but it's still early days. The limitation on multiple genes is just that, I think you start squaring or cubing or, basically what are the chances that you get both copies of the gene in the right location that are both expressing? And there's just not many diseases that are resulting from the knockout of 2 genes. We just don't know. Most of the rare diseases that we see are almost all single, you would have to inherit 2 negative copies, right, from both parents. So the chances of those kinds of things get really low. So there's actually not much therapeutic value right now from what we know of adding 2 genes. Does that make sense? Yeah.
You're saying that you eliminate all your the stem cells from a patient and you replace the ones that are you change the ones, and that's how you generate
and so that's how modern immunotherapy works too. So you probably have heard of companies like Kite or Juno that have basically been pioneers in the new space of engineering immune cells to go after cancer, and it's the same kinds of things. Although there you don't have to eliminate the immune cells, you just grow those up and then re-inject them through infusion over time. So you don't have to do a bone marrow transplant in those cases. Yeah.
I was wondering, so I understood the reason for immune diseases, but why for disease of the eye is this also being used?
Because another viral vector, there's basically the eye is a little bit immune privileged, so you won't have a massive response to certain viruses, for instance, like AAV. So you can actually use AAV delivery fairly effectively in the eye. So people are using it. So a lot of the early companies in this space, like Editas and others, are trying to fix diseases in the eye as their entry point initially, because it's going to be the easiest. Does that make sense? Okay, so I think so. Let me just zoom out for a second. And kind of tell you where we're going with Octant. So so one thing I'll say is I am super excited by these spaces. I think the ground has shifted so much in biology over the last 5 years that it's insane what you can do now that you couldn't do 5 years ago. But I think where the real magic that's going to happen is not in we're going to CRISPR away every disease. You're never going to CRISPR away hypertension, both for scientific reasons, but also just from practical reasons. You're not going to take drugs to cure most of the diseases that you know that are going to be CRISPR-based. Mostly, this is, for a long time, at least as far as I can tell in the future, going to be, and we'll talk about this a little bit, but are going to be rare diseases. So that's one thing. The second thing is, I think these technologies are changing things in pretty profound ways. Not just in human biology, but also in the engineering of the living world. You probably know of companies that are using synthetic biology to build the set of industrial chemicals and foods and other things that we interface with life and work with them more rationally. So I think that's all very exciting. I think the challenge with a lot of these things, though, is that there's a suite of these technologies. They've all changed the face of each of their own kind of practitioners in their fields. But I think where the magic is going to happen is when you start thinking about how all of these changes you can be used to reimagine drug discovery and how we can combine these tools in super powerful ways. And so the intersection of these fields that are all changing, plus computation, which is also changing, and our ability to interpret many of the perturbations we're making, I think it's going to be an incredibly exciting time over the next few decades in biology and in science, and hopefully in medicine. The ideas behind Octant really came about thinking about what the future might look like for how we treat diseases, especially diseases that we all know about. And so to understand that, maybe I'll pull up a different slide. I guess I'll have to tell you a little bit about human diseases and human traits in general. So what I've been saying for a long time is that there's different ways to there are different lenses in which to think about genetic diseases in which they arise. The rare diseases that I've been talking about, these include like a ton of syndromes that we talked about, but like thousands of syndromes that you've never heard of that only exist in like doctors and patient rooms. And these are very small cohorts of people, 10 to 100 to maybe 1,000 people that have this disease. We're starting to get better identifying that because we're sequencing their genomes. And we're also getting better at treating it because we can do things like build patient-specific stem cell models and try to repurpose drugs or do CRISPR to fix those changes, because usually the effect, as I mentioned, is due to a single large effect mutation that knocks out a gene, right? And so, or two in the same gene. And that's interesting and powerful, and there's a whole new set of ways to monetize that, which has driven a lot of excitement in this space. So there's this thing called Orphan Disease Act, which allows for the monetization on super small cohorts and the advancement of clinical trials in those areas. So you can charge people $1 million a patient for these types of therapies, for better or worse. Somatic diseases are diseases that arise due to mutations that arise during your lifetime. So these are things like cancer. Again, these are driven by like usually large effect mutations that cause a deletion in a gene that might suppress tumors normally, and then this causes kind of either through single hit or multi-hit models causes the evolution of cancer in your body. There's a lot of excitement and really cool data around a set of therapies called immuno-oncology, which is to harness the immune system to go after those, to recognize and go after and kill those changes. And that's been super promising and lots of excitement, a lot of companies and a lot of development in that space. And that's exciting because you're building an evolving therapy, which is your immune system, against an evolving disease, which is cancer. So all of that makes sense. Every disease that you probably have heard of and probably know people that suffer from, so every neuropsychiatric disease, every metabolic disease, like diabetes, schizophrenia, depression, these have a very different genetic basis than these prior two categories. And the genetic basis of these are called complex traits because they're genetically complex. They're usually the inheritance of hundreds to thousands of what are called common variants that all give you small effect size accretion towards susceptibility towards the disease that slowly push a particular cell type or pathway into pathophysiology. And so treating them is super hard, right? Like you can't just make thousands of changes across the genome. We, A, technologically can't do that, and B, there's no way we could do that to adults when most of these diseases manifest. And secondly, we don't understand it that well, frankly. And so the way that traditional drug discovery happens in this space is around like basically phenotypic responses, clinical trials of small molecules or other targets that hit these kind of central nodes in physiology in human physiology that are known to like affect lots of other things. And the hope is that we can use these things as a way to kind of get at new medicines. At Octant, what we do is basically combine many of the technologies I told you about to basically, and I'll skip over much of this, but I'm skipping over. Sorry, something. But where is this? We'll start with here. The types of experiments we do at Octant basically combine the set of technologies that I told you about. And so in a cell, and so there's trillions of cells in your body, there's different cell types. Each cell type has a prototypical set of genes. And there are things called receptors, and receptors are actually just targets of small molecules, and they change their activity depending upon the activity of a small molecule or hormone or peptide. And these types of receptors are kind of classically the way that we kind of get a handle on physiology, right? So, or as a way of altering physiology. So these cells normally have these receptors on the surface of the cell, and they're basically the cells are using this to like sense the outside world and respond in kind of reasonable ways to insulin is one of these kind of hormones that will receptor have the cell will respond to in rational ways, which is either uptake or release of glucose depending on levels. So what drugs do is hijack those receptors as a way to, when things are going wrong, as a way to like modulate their activity in kind of physiological ways. What we do is try to engineer cells so we can report on the activity of receptors. And all that means is if you have a receptor in a particular cell, if it gets activated by the action of a small molecule, we add like essentially a print statement using CRISPR and other editing technologies and synthesis technologies, and we print out a barcode. And that barcode basically is something that we can measure by next generation sequencing. And the printing of this barcode is literally the transcription of the barcode from DNA to RNA, and we measure the levels of RNA of this barcode. And what this barcode will tell us, the number of counts of these barcodes from the sequencing experiment, tells us how much this receptor is activated. Right? The cool part about this is you switch what usually in the past has been a light-based activity. So normally the basis of most pharmacological discovery efforts are what are called luciferase assays. So instead of producing a barcode, you produce a gene that makes light, and that light will light up and you can detect it. The problem with light is that for each new thing that you want to make, you have to do a different test. What we do is make hundreds to thousands to millions of these basically engineer genetic changes in human cell lines, use like genetic engineering to edit these human cell lines, use synthesis to build these barcodes and circuitry to report on the activity, and then we use sequencing as a measurement device rather than a device, like a counting device for each of these barcodes. So what that lets us do is in a single experiment, instead of measuring one output, we can measure hundreds to thousands. And what we try to do is be as comprehensive as possible. And we can tell you more about the biology and the types of strategies that we're going around, but that's a basic fundamental point, is that we're basically hacking biology, which is a super complex machine that's like in all essences an alien technology, and we're trying to figure out like what are the hooks that we can press to turn on and off things. And if we're interested in building small molecule drugs or drugs in general and trying to be rational in this space, it's crazy to think for me that in 5 or 10 years we're going to be in this place where we're not measuring how these small molecules are interacting with every target of human physiology in the genome. And in the end, there's 20,000 of them, which is like a countable number. And so the fundamental theory and hypothesis of Octant is that we can build this set of systems that combine these technologies along with computation, and I'll talk about that next, as a way to map how chemicals interact with the target of human physiology writ large. That's the hypothesis, right? And so what, from a computational standpoint, we're maybe almost everyone at the company is kind of dual, but we're looking for some of our first software engineers to really help us scale and think about these problems where we generate massive data sets between. So what we've done in Octant over the last couple of years has been scale this type of work towards a class of receptors that are called G protein coupled receptors. They're basically the targets of, the biggest class of receptors in the human genome. There's 800 of them out of the 20,000 genes. 400 mediate our sense of smell. Another 50 are sense of taste. And then the other 350, basically every major physiological process in your body. So they're the targets of more than half of all prescriptions, a third of all FDA-approved drugs, pretty much every illicit drug that you've ever heard of targets these receptors. And they're just really good modulators of human physiology. The challenge is that you're usually taking one of these light-based luciferase assays, building one GPCR and running a small molecule screening campaign against these things. What we're trying to do is be much more comprehensive about that in the anticipation of, one, building better drugs, but two, building safer drugs. So there's many cases of off-targets on GPCR where you're building a drug for one GPCR and it hits another GPCR, which is not surprising because all of these things have shared evolutionary ancestry. They all duplicated and diverged from the same set of things, and they're oftentimes sensing the same thing in the body. You're trying to get specificity without having any view of that specificity broadly. And so what we're trying to do is build the data sets that will allow us to be more rational from how to relate chemical moieties to the activation of these types of receptors. And you probably know of many big cases when that's gone wrong. So Vioxx, Fen-Phan, these are all big examples of off-targets against GPCRs or GPCR ligands where we just weren't looking for those off-target effects, but chemicals affect lots of things. And so trying to be rational about how chemical moieties interact with the targets of human physiology as a way to be generative about new molecules that will be more rationally better towards disease. So that's what we're doing. From the software and computational standpoints, we have some not severe, but major, and I think exciting problems to work on. One is that there's a massive amount of computation that goes from turning the sequencing-based assay into a real assay that can measure the effect of drugs on targets. And then two, the sets of data that are coming out, both from these types of assays, but also kind of the large public data sets that are coming out of where are these receptors, which cell types are the receptors on, which combinations of cell types do we see, which combinations of receptors do we see per cell type, basically converging that with the molecular phenotype data that we're getting that are on much more massive scales, orders of magnitude bigger scales than has ever been seen before, and in ways that are comprehensive. And can we use that to build tools? And so we work with a class of machine learning tools that are being developed and are developed, and some they're kind of all over the place at this point, but around relating chemicals and chemical moieties to these kind of large-scale data sets. And how do we use that to be generative about new types of small molecules that we want to go create? So that's a proposition, and that's what we're working on. I think that's all I really had. I'm happy to jump into more details on stuff, either more generally or more about Octant.
Apologies if I just missed this, but this is all in vitro, in the lab?
Depends on what you mean by in vitro. These are in human cell lines.
Yes. Okay, I see what you're saying. Yeah, so it's in the lab at least.
Yeah, it's not. We're not doing this in people.
Right. Okay. I mean, so this isn't. Is there a?
We're not genetically engineering people to act as reporters for adult drug development.
Well, I mean, but you could imagine wanting to know where you are, what the expression is of drugs of particular things in your body.
Exactly. Yeah.
But that's not in scope for the moment.
It is in the scope, but the interesting thing about that is that there are a bunch of very large-scale projects to do exactly that, which is to map every cell type in the human body and the expressions of the genes in those cell types. And all that data is public and released as it's generated.
Right, but that's different. That's expression of genes where you're talking about receptor,
Sorry, these receptors are genes in the human genome.
Okay, this is only, sure.
Yeah, does that make sense?
Yeah, I think so.
So there's 350 of these GPCRs. We're definitely interested in humans. Every cell type probably has around 20 to 30 of these expressed. We're super interested in the combinatorial aspects of those because, and we can get into this more, for complex diseases, this idea that you're going to hit a single target hard works some of the time, but the big challenges aren't trying to find a single receptor that we're trying to hit. The big challenge is that if you look at the best-in-class drugs for super complex diseases like schizophrenia, depression, obesity, all of those drugs are what are called dirty drugs and were discovered by accident, and they hit 20 to 30 receptors. So things like atypical antipsychotics, things like lithium, things like metformin, things like marijuana and pain. All of these kinds of things are very complex. That's either a single molecule that has very complex physiology or many molecules that you can't just isolate one of them and have it work. And the basic hypothesis there is that you're not achieving specificity by a single receptor on a single cell type that might be in your liver, because like for a lot of these receptors, so like one of the receptors we worked on in my academic lab is called the beta-2 adrenergic receptor. It's a target of albuterol and things in like asthma inhalers. It opens up, it's a receptor in the smooth muscle cells of your in your epithelium, your airways, and if you agonize it, if you turn it on, it opens up those airways. So that's why it's a good asthma medication. That receptor exists in your heart. It also increases your heartbeat. It exists in your brain. We don't know what it does. It exists in every major organ system of your body, and it does different things in those different cell types. And so one of the fundamental hypotheses that we're very excited by is by having these maps, and because these pathways synergize on each other, if you can hit the right set of receptors that achieve specificity through this combinatorial nature, rather than hitting a single receptor, you can achieve specificity on a particular cell that's going wrong, rather than a particular receptor that's going wrong. And if you think about the genetic basis of complex diseases, it's not a single hit target, it is this more complex set of things. So this idea of engineering polypharmacology and being rational about polypharmacology, because all those drugs were found by accident. Like atypical antipsychotics were fertilizers, right? Like, or pesticides, I forget which one. But like these things were discovered kind of phenomenologically a long time ago, and the efforts to make them more specific have not worked. And it's a fact that polypharmacology that's important.
I guess the reason I was asking about in body is like you might have specific, like this is I guess the personalized medicine, which is I guess different from what you're talking about, creating, let's say maybe cocktails or creating
Molecules that have
that have particular properties that generalize across people. Is my understanding, unless you're taking cell lines from someone and then learning from that. So I'm just curious if this is also applicable to that, or if that's sort of beyond the scope of
No,
this
it is. So like some of the stuff I didn't mention, and so like we have an experiment on that beta-2 adrenergic receptor. So where, like I mentioned, we can combine engineering of cell lines, synthesis, sequencing. So what we did in this experiment in my lab is that we made every single possible mutation to this gene. So there's 400 amino acids, there's 20 amino acid choices at each position, that's 8,000 different mutations. We covered this at 100-fold. We covered this at 50-fold coverage, so we have about 300,000 to 400,000 cell lines, and we can agonize that with FDA-approved drugs and measure the consequences of all of those mutations in a single experiment. It's interesting. It's a really cool research project. It's a hard... we are very interested in going after these big disease targets that are like basically running out of options. So there's like everyone in pharma is moving towards oncology and rare disease. But complex diseases are basically every disease that we know, when we all have people that we know that suffer from these things, and like there's been very few new approaches in this area. Why we're excited by this is we know genetically that why these things occur is because you inherit a set of small effect mutations that drive a particular cell type into pathway. It's everything this genetics is telling us it's this combination of mutations that lead to these things of what are called common variants. So you inherit an assortive number of a set of, so every person has about 5 million deviations from reference. The reference genome, you have 3 billion bases or 6 billion bases, about 5 million changes from the reference genome. 99% of those are common variants. So we all share that. That all those mutations existed since out of Africa expansions. Like they're old mutations that existed throughout the human history. These rare mutations are things that arise during our population expansions. And so it's kind of why people say, well, we're mostly similar, 99% similar, right? Because we mostly share the same variants. And these common variants are what are thought to drive susceptibility towards common diseases. And it's these rare variants that lead to the rare diseases.
Will Octant build and sell software and services to drug companies, or will you become a drug company?
We will become a drug company.
Cool.
Yeah. Obviously, I say that with like the massive amounts of humility of what a drug company does, which is much more than early stage drug discovery. It's going to take a long time for us to get there, but that's the intention. But I think I think in general, the way I think about Octant is that it's a series of things that are coming together to go after a problem that I care about and want to go after. I also want to build Octant in a way, and we care a lot about building Octant in a way that's actually helping the community in general have the tools and the developmental systems to be able to think about biology in this way. So we will release stuff. We probably won't be charging for it, if that makes sense. So those are kind of released through code and publication. So like we've had to develop a whole synthetic biology pipeline to deal with the thousands of cell lines that we're engineering. And like one of the things that we're thinking a lot about is like just releasing that to the public to enable the community in general to have access to those things. But I think that would be more from trying to do good things, yeah.
So can that allow you to target a specific cell in a specific location in your body, like for example, by targeting receptor A and receptor B, but not receptor C? Like
yeah, that's the idea.
Boolean logic.
But with the engineering of molecules, essentially.
Mm-hmm. And so drug companies don't do that at all?
They do. The best, like I told you, the best-in-class medications for most of these things are like either combination drugs or polypharmacological drugs. But like, they're almost always discovered by accident or by combinations of existing drugs rather than being rational about why you want to achieve polypharmacology. Secondly, I think this kind of multimodal discovery is new. Like we haven't had this ability to kind of look at, instead of individual data points, vectors of data points that are very large for each of these chemicals, and we can do this at pretty large scales. And so the idea is that combined with the computational capacities of making sense of that data, which has only also been developed around chemical learning tools like graph convolutional neural nets and autoencoders that encode like kind of lower dimensional subsets of chemical compounds as a way to be generative around what are the next 100,000 chemicals I should run through this assay. So this like turn the crank biology. We can do this. We do this right now with hundreds of receptors in a 10,000 chemicals at a time once a week, right? That type of ability is just crazy. And so given this new perturbation and capacity to design, build, and test these molecules, I think we're hoping, the bet is that we can be a lot more rational about building molecules that do these kinds of things. Yeah.
Can you talk more about the computational challenges? For instance, you mentioned you have large data sets. So how large are the data sets and what are you doing with them? Yeah,
so I think there's like the convergence of data sets, which I think there's a lot of big data stuff that like a lot of people are doing. I think the really technical challenges are, how do you think about convolutions in chemical space? Right? How do you make chemical space differentiable in order to basically leverage the powerful tools that have been brought to bear in the reinforcement learning and active learning communities? I think there's a bunch of ways to answer that problem. In my mind, I'm a little agnostic. I can tell you some of the ways that people have been thinking about them. One has been, let's just take chemical. So there are word-based representations of chemicals. They're called SMILE strings. It's a rule-based set on how to describe a chemical in a series of letters. In theory, you could just use the same word-based tools that have been developed in the machine learning communities around the representation of words and use that as just basically like convolutions of words or long LSTM, wherever that is. I forget, long short-term memory based approaches. And so there's one approach. Another approach is like even simpler. There's pictures of molecules, right? There's defined ways to do that. You can actually just use image-based convolutional neural nets. There's the more advanced approaches that are trying to actually improve chemical properties into these systems. So there are autoencoder-based methods to like bring chemical space into a lower dimensional subset of a latent space, and then use that as a representation, as a way to like basically compress the data. So this is kind of like development of autoencoders, and if you can do that, distance in that space, even though it's high dimensional, can be a representation for things that you should build next. Obviously, and then there's the graph convolutional neural nets. So like thinking about convolutions and graph in connected spaces. There's been a bunch of recent work on that. Those are kind of the approaches that are out there, and there's more. The interesting thing, I think, is to take those frameworks in the context of a reinforcement learning, a batch reinforcement learning based approach, which is what we have. We're not going to go, chemical space is infinite, essentially, beyond a certain number of atoms. And so we can't do all of those. But what we can do is turn that once a week, 10,000 chemicals at a time, through kind of easily synthesizable spaces, like chemicals that we can access through standard chemistries. And what we're very interested in is like, how do we traverse that nearly infinite space in order to get the sets of activities we want? So what we're very excited from a deep computational standpoint, not just like pure software engineering stack, but like a deep computational standpoint is like, what are the approaches that are going to get us there? We have thoughts on that, but there's still very early stages on that. Does that make sense? So that's one of the computational approaches. I think there's a whole statistical framework that we've been working on, and I think we have somewhat of a good handle on because we've been doing these. We were kind of the people that have developed these experiments for the last decade. Not just these receptors, but these kind of what are called multiplex reporter assays in general. And there's a whole bunch of computational challenges around interpreting and understanding large-scale data sets like this. And then just finding like, I think like in every computational approach, you have, especially ones that deal with physical systems, you have to be a little bit true to the physical medium in which you're dealing with. And so they're not purely computational challenges, but a challenge of diving into the physics and biology of those systems. And that's hard, right? And it's hard to get people really excited in physics and chemistry. But I think if we want to interact rationally, like we have an academic project in the lab that I'm stopping because I'm stopping my academic lab, but like it's a very exciting, well, we've done this with a fairly simple system. So in bacteria, there's this thing called bacterial promoters. They turn on transcription, which is DNA to RNA, which kind of is this whole field of gene regulation. It's how this bacteria sense and respond to systems. They're on the order of 100 nucleotides long, but there's no predictor of what a promoter looks like. We have basic ideas of the mechanism. We studied it for 50 years, but if you gave me an arbitrary DNA sequence of 100 nucleotides in length, I couldn't tell you if it's a promoter or not. Seems like a simple challenge. We built a system where on every 2 weeks we can turn 100,000 of those experiments where I can completely design the sequence and measure the quantitative response of that promoter. So seems like a simple problem, but it's not, because 4 to the 100 is 2 to the 200, which is 10 to the 70, right? So 10 to the 70 is much bigger than you can ever compute. And so then the challenge becomes like, how do I think about establishing causal mechanisms or building generalizable predictors? And this is kind of a class of machine learning around reinforcement learning, but also in statistics around like design of experiments, things like, there's a whole class of experiments called the multi-arm bandit problems where you can use kernel methods like Gaussian processes to establish uncertainty of where you're most uncertain, and building the next set of sequences that are most uncertain. So like, how do I think about basically rethinking about the scientific method given this new power of the ability to manipulate biological systems and measure them at scale? I think it's a super fascinating problem. We're doing that in a way that I think is more monetizable than bacterial promoters.